Okay, I think for the interest of time, we will start. And I just wanted to say a very good afternoon to everyone joining us for the first Revive webinar of 2026. We look forward to some insightful presentations throughout the year and I invite you to please share the invitations that you receive within your own networks. Just some general housekeeping rules. Please keep your microphone muted during the presentation as we will allow a question and answer session at the end, but you're also welcome to pop your questions in the chat box as we go along. Professor Graham Maintjies, the chair of the Revive Steering Committee, will start with a short study update, and I will hand over to Graham now.
Thanks very much, Elsa, and welcome to everyone. I'm just going to share my slides that I've got prepared. So for colleagues joining us for the first time, these webinars, we always present a 4 or 5 slide update on the progress of the Revive trial on behalf of the trial leadership. And we don't assume that everybody knows about the Revive trial. So the first slide just always covers a brief overview of what the trial is. So the Revive trial is a randomized placebo-controlled trial being conducted in multiple countries across Africa, funded by the Gates Foundation, evaluating an intervention in people with advanced HIV disease. And that intervention is azithromycin prophylaxis for the first 4 weeks of commencing or recommencing antiretroviral therapy in adults with a CD4 count equal to or less than 100 and no acute illness requiring antibiotics at the time of entry into the trial. So participants receive either are randomized to either azithromycin or identical placebo for 4 weeks, and then are followed up for 1 year. The primary endpoint is at 24 weeks and is of all-cause mortality. And our hypothesis is that among patients with advanced HIV and CD4 of less than 100, starting or restarting antiretroviral therapy, this intervention will reduce mortality compared with placebo at 24 weeks of follow-up. And all participants are provided background standard of care for advanced HIV disease, which includes antiretroviral therapy and other WHO recommended interventions. So the trial is currently enrolling in 11 countries, shown in green, and is planned to start recruitment in an additional 2 countries in March this year once approvals are in place. And there are currently 57 sites active and is led in a partnership between the PHRI, Canada, and University of Cape Town. So just an update on recruitment. The target sample size is a total of 8,000 participants. And late last year, we passed halfway. So there's currently 4,537 participants enrolled in the trial, and just over 200 in the last 30 days. And what's clear is that all countries involved are contributing in an important way to the very impressive enrollment into the study. And there are 3 countries where enrollment is around 1,000, and that's Nigeria and South Africa and Uganda. But all countries are contributing to the enrollment, to the trial, ongoing enrollment. And then the final slide, just to show you the projected enrollment, recruitment projections. The blue line is what we estimated at the beginning of the trial, and you can see that the purple line is actually showing that recruitment is over what we had anticipated. And apart from the period around the end of last year and the festive season where recruitment decreased, we were recruiting around 300 participants a month across all of the sites. And if we continue with that, we estimate that the trial will be fully accrued by early next year, so early 2027, but obviously follow-up will continue for another year thereafter. So just congratulations to all of the sites that are contributing to the very impressive enrollment and follow-up of participants and collection of data. And with that, thanks, Elsa. Back to you for Ruan's talk.
Thank you, Graham. It is my privilege to now introduce to you today's speaker, Dr. Ruan Marais. Dr. Marais is a clinical microbiologist who holds fellowships in medical microbiology and virology from the South African College of Pathologists. He completed his MSc in virology at Imperial College in London and has MMed degrees in virological and microbiological pathology at the University of Cape Town. He's currently completing his PhD in bioinformatics at UCT. Dr. Marais is a CRIC Africa Network Career Acceleration Fellow and a contributing investigator at CIDRI Africa. His research focuses on developing novel molecular diagnostics with infectious diseases with a particular emphasis on the software needed to translate these technologies into clinical care. Dr. Marais will be presenting on the detection of antimicrobial resistance with limited resources. And with that, I would like to welcome Dr. Ruan Marais to today's talk.
Thank you. Just share. Are we all good with, does it look fine, the presentation? Can everyone see?
Ruan, it's not in presenter mode. We're seeing that.
Let me see.
No, it's.
How's that?
That's good. Yeah.
Perfect. All right. So I'm Ruan. I'm going to be presenting on AMR in limited resource settings, and particularly around the issues around detection of AMR or the lack thereof. So just to contextualize first, there's a paper published in 2024 that evaluated the global burden of AMR between 1990 and 2021. Really impressive study with a vast amount of data and a lot of modeling work done as well to forecast the burden up to 2050. And from this study, it's really clear the burden that AMR presents, with a forecasted 1.91 million attributable and 8.22 million associated deaths expected by 2050. Some important things to note from the study or data that can be extracted is that in children, in particular, Klebsiella pneumoniae, strep pneumoniae, and E. coli are the important pathogens. And for all groups, the largest increase in attributable burden was MRSA, doubling or more than doubling from 57,200 attributable deaths in 1990 to 130,000 in 2021. And then among gram-negative bacteria, resistance to carbapenems increased more than any other antibiotic class, notably from around about 127,000 attributable deaths in 1990 to 200,000 in 2021. The paper also went further and suggested potential reduction in mortality or attributable deaths with specific interventions such as IPC, which they predicted or modeled could prevent at least 300,000 deaths, and pediatric vaccines, which could prevent an additional 181,000. Now that's the overall study and the backdrop, but really if you look at the actual data, two major things stand out, and that is firstly that not unexpectedly, sub-Saharan Africa was the super region with the highest mortality rate attributable to AMR in 2021. But more importantly, the study's main limitation was the scarcity of data from some LMICs across the board in terms of the data points that they were evaluating, such as distribution of pathogen by infectious syndrome, relative risk of resistance for key pathogen drug combinations, and the number of deaths related to infection. And you can see here the mortality rate in 1990 in the top figure, and a noted improvement, particularly in pediatrics in 2021, but with data scarcity being the primary issue. And if you look at the actual numbers, it really is clear the disparity in data available. So for example, looking at Central Europe, where in the order of tens of millions of data points are available to make these assumptions and generate these models, versus Central Africa, where there's a few thousand data points included. And that trend holds across Western Europe, where 100 million data points available, versus Western Sub-Saharan Africa, where you only have about 115,000 if we're purely looking at the number of deaths. And the data is far less if you look at the more rich data, such as sepsis, infectious syndrome, specific data. So really, it's clear that all of these findings, so Klebsiella, strep pneumonia, E. coli, if we now revisit what I mentioned in the first slide, all of that, making that assumption is dependent on sampling, culture, and identification, making the observation that the largest increase is due to MRSA. That again, now you need AST as well, antimicrobial susceptibility testing. Talking about gram-negative carbapenem resistance, you have to extend that further, where it's sampling, culture, AST, and in most cases, molecular testing for enzymes to really make decisions on how to appropriately manage cases. And then the suggested actions in particular, so suggested infection prevention and control, that requires, again, laboratory infrastructure. For audit purposes, you can't put in place interventions if you can't audit outcomes. And pediatric vaccines, that requires significant infrastructure in performing typing such that vaccine strains can be selected. So really, it's clear that the lack of data is reflective of the absence of this infrastructure in low middle income countries. And that lack in this study really highlights what aspects of infectious disease practice and medical microbiology are absent. Because if we can infer that these resources are not available, then we also have to assume that culture-based antimicrobial susceptibility testing followed by targeted therapy is not broadly available. Inappropriate therapy potentially is a frequent occurrence, because life-threatening infections like non-typhoidal salmonella and Plasmodium falciparum infection are potentially clinically indistinguishable. Further stewardship, such as optimization, de-escalation, and discontinuation of antibiotic treatment, that is not possible without clinical laboratory service. And aggregated susceptibility patterns to inform empirical guidelines can't be done, nor can outbreak investigations occur. So we can really infer that the reflection of that lack of data, indicating the lack of laboratory services, has incredible knock-on effects on what is available and what can be done in infectious disease practice. And it's really a reinforcing cycle where you have reduced infrastructure, people, and resources, resulting in less research and routine data, which is usually used to generate things like the WHO priority pathogen list. That policy is used to allocate funding, which is used to produce infrastructure, employ people, and make resources available. So really, what we think is important is potentially not necessarily important, or it is not appropriate to what we need to focus our efforts and our research on. So just as a specific example in action, the WHO priority pathogen list is focused on carbapenem-resistant A. baumannii. The ability to isolate and assign a pathogen as a carbapenem-resistant A. baumannii assumes the facilities or infrastructure to identify the organism, to do the susceptibility testing, are both available. Thus, you can potentially indicate this is a lack and thereby access funding, or if you identify this as a particular problem in your hospital. The same with carbapenem-resistant Enterobacterales and third-generation cephalosporins. If you're unable to isolate these and specifically identify and include this in your data, it's very difficult to to write into a grant or approach an NGO or develop a national plan because that data is unavailable and can't be actioned. Another example, an example would be MRSA, where this study clearly indicated the greatest increase in attributable burden is from MRSA. But that was not put as a critical priority pathogen, specifically because it was noted that it has high treatability and medium preventability. But that preventability assumes that an antimicrobial stewardship service is in place, contact precautions for colonized or infected individuals with MRSA can be implemented, and MRSA screening and surveillance can be implemented, which, if you don't have a functioning clinical laboratory service, are none of which is possible. So it really highlights that our lack of data is a primary problem in terms of improving care, because if we don't have information about what we are experiencing as problematic pathogens or emerging outbreaks, then that makes it very difficult to assign these as critical priority pathogens, which makes it more difficult to access funding and resources to address the problem. So really, the question is, why is it such a challenge to produce this report? That it would seem relatively simple. Like you take a sample, you put it on a plate, and if it grows, if it doesn't grow, the antibiotic is susceptible. If it does grow, it's resistant. That seems really quite simple. But what is underappreciated is that producing that result is quite challenging, because it is essentially a tiny area in which the results are repeatable and reproducible and trustworthy. So, for example, if the culture result, if the temperature is different, that may impact the recovery of the organism. If the time to receipt at laboratory changes, then that may impact the result. If different media types are used, that may impact the results. And while that is certainly still a problem in clinical pathology in general, with something like hematology, there are markers that you could potentially use to gauge the quality of the sample that you've received. So, for example, the amount of hemolysis. But in clinical microbiology, the major overarching problem is that we're trying to prove a negative. So does no growth indicate that there is no infection, or was it because our organisms lost viability? And that leads into probably the major issue and why clinical laboratory services are not as readily available, because you need an overarching quality management system to control that data generated so that it's reliable. And that quality management system has multiple components which have varying levels of costs and complexity and required infrastructure. So while at the very basic level, standardizing inoculars, so purchasing equipment that can be used to measure optical density, for example, can improve standardization and is necessary for quality results. There are far more expensive and complicated aspects that also need to be considered, as opposed to a chemical analyzer where you could purchase a blood gas machine and produce a large number of relevant tests with a single instrument and a single supply chain and a single service engineer. So things like organism zone measurement, if systems are not in place to for example, highlight differences between different organisms, so let's say trimethoprim sulfamethoxazole, where organism inhibition is rated 80% versus complete inhibition, that can alter the results produced with potentially knock-on effects to public policy, as well as empiric regimens for various organisms. Incubators need to be held at specific temperatures and at specific atmosphere conditions. So does your lab have a standard supply of CO2? Media is a perishable, And for example, if the wrong salt concentration or the wrong agar concentration is used, then the zone size of inhibition is going to change and may have knock-on effects on your produced results. Then multiple different instruments are required for organism identification, be it APIs, MALDI-TOFs, Vitex, each of which require consumables, service engineers, and are quite expensive instruments. Then biosafety can't be ignored. You can't safely provide results if there is a risk of your laboratory technicians acquiring infectious diseases such as brucella. You need biosafety cabinets where appropriate. And then overarching all of that, a quality management system that ensures that the media quality is good, that the incubators are kept at a specific temperature, that your instruments are serviced, and that your biosafety cabinet has passed the smoke test. And in that, you have documentation, validations, verifications, and running that or leading that, you require sustainable technical staffing to ensure that the quality management system is implemented and maintained. And of course, you can't necessarily just deploy an NGO that has temporary staff, provides a service for a trial or for a specific amount of allocated funding. But when that disappears, the staff return home and the service disappears, and that may impact how our hospital can function if now they are reliant. So there are various resources that laboratories can use to go along that process to become validated. So for example, many clinical laboratories are validated according to the ISO 15189 standard, or the CLSI QSE tool that was developed from that, where multiple aspects are evaluated or included to ensure that a laboratory produces quality results. And that goes all the way from how the institution is organized to maintaining or establishing stable purchasing and inventory systems to actual laboratory assessments and external quality assurance and internal quality assurance procedures, down to a culture, or not a culture, but a requirement for continual improvement and a system for dealing with non-conformances when policies or SOPs are deviated from. And the reason all of that is necessary is because every single bacteria is going to require different conditions to be effectively isolated without bias. And the conditions for a strep pneumonia might not be the same as an Neisseria species, might not be the same for syphilis. And the challenge can be seen even in very well-funded large trials that occur in arguably optimal conditions. So, for example, the DoxyPrep trial, which looked at post-exposure doxycycline to prevent bacterial sexually transmitted infections. Now, you might assume correctly that knowing the susceptibility profile of the Neisseria gonorrhea species in your trial participants is quite important. But only about half of participants were able to provide a sample, and only about 10 to 15% of those cultures actually grew. And that's because Neisseria gonorrhea is very difficult to grow. So a trial with hundreds of participants only having maybe 5 to 10 cultures of the organism is potentially problematic. So looking at the REVIVE trial specifically and the AMR sub-study, what have we done to look at those risks and those potential biases? So certainly the AMR sub-study, where we'll be sampling multiple biological samples, is not going to be placed in an optimal clinical microbiology environment. There's not going to be a sample that is transported to a lab in the same facility, processed within a few hours at most, interpreted on site, and added to a laboratory information system in an ISO accredited lab. So how do we deal with that bias? How do we deal with the risk of lost pathogen viability? Well, in that instance, we designed a custom culture media that specifically, as opposed to just STGG, which is the WHO recommended culture media for strep pneumoniae, we needed to expand that because strep pneumonia is not the only pathogen that we're looking at. So STGG media, we found in initial testing that while that does reliably stabilize and result in recovery of strep pneumoniae, it alters the pathogen distribution for other organisms, such as E. coli, where you have replication of the organism within the sample, which is really what you don't want in a stabilization transport media. You want to maintain the relative concentration of organisms until you get to the point of analysis. Then we also will be doing culture as well as molecular testing, because some pathogens you may lose viability. For example, in the doxypep trial, that was a primarily molecular first testing approach, where nucleic acid amplification testing was done, followed by culture. Then how do we reduce the bias due to pathogen natural history of infection and colonization? Individuals may not be persistently colonized or maybe intermittently colonized with different organisms. So there we've tried to optimize multiple sampling time points and sites to optimize our chance of recovery of relevant pathogens such as Salmonella, Staph aureus, and Strep pneumoniae. And then how do we reduce the risk of bias due to selective enrichment, as I mentioned with STGG, or depletion of specific pathogens by the media? I mean, therein we do therein validations of media as well as molecular techniques are really important. So, for example, we tested before sending out the first sample containers, we tested the viability after 7 days in freezer storage, both E. coli as well as Strep pneumoniae, comparing STGG, which is the WHO recommended media for Strep pneumoniae, with modified AMR media, which is more appropriate to recovery of a large range of organisms. Because STGG includes glucose, which is going to result in organism replication, while modified AMRs specifically is only a buffer solution that maintains viability but does not allow replication. And then we modified the AMRs by adding glycerol to allow us to freeze our isolates immediately, as opposed to standard AMRs media, which that is not possible for. In the validation, we found similar performance to these two media types, but that is ongoing at multiple different time points. Right. Then other really important aspects to why a quality management system is important in maintaining trustworthy and reliable results is that guidelines change. So interpreting susceptibility of an organism based on guidelines from even a few years ago. So, for example, looking at Kefapime CLSI criteria from 2013 versus 2026, if an isolate was tested a few years ago, it's with an MIC of 6 or 4, arguably, you would have called it susceptible, while now it would be called intermediate. Same with amikacin, where the susceptible breakpoint has changed from 16 to 4 in the 2026 version. So if susceptibility results are reported based on criteria from an old version of the CLSI guideline, for example, then these results may not be comparable to results that are reported based on newer versions. And as we know, selection of empiric regimens based on reported susceptibility results is a primary activity that is done. So if that is incorrect, that may result in multiple patients receiving incorrect therapy. And then another example would be what the actual interpretation criteria mean. So, for example, a few years ago when there was a diphtheria outbreak, these isolates were sent to a lab that used UCAST criteria for susceptibility testing. And in UCAST, the eye does not mean the same thing as intermediate in CLSI, where specifically it means increased exposure, not intermediate. And that and that can result in misinterpretation. I think someone has a drawing on the screen. I don't know if so understanding what the interpretation criteria mean is of primary importance, because if you report, for example, diphtheria as penicillin I, under UCAS, that means penicillin increased exposure, and there is no susceptible breakpoint for that organism. And that can result in the perception that there's increasing rates of resistance, when in fact it's just a artifact of how that antibiotic susceptibility is reported. Then, notably, organism identification is extremely important. So even within a genus, such as looking at Staphylococcus specifically, a Staphylococcus epidermidis, which may look exactly the same as a Staphylococcus schleiferi or a Staphylococcus lugdunensis, different breakpoints are used for susceptibility testing. So it is extremely important to have a identification system that can speciate specific pathogens to be able to reliably report susceptibility results. And that's why it is somewhat problematic that, for example, in this systematic review, only about 5% and 6% respectively of laboratories reported ISO accreditation and external quality assurance in reporting AMR data, which is compounded with the fact that 42% of countries in the African continent had no data available. All right, and then a further study looking at bacteriological testing and AMR detection in 14 countries, only 1% of laboratories in the region deliver bacterial testing, of which 14% are also not capable of conducting AST, despite urine analysis, microscopy, blood culture, pathogen identification, and AST all being included in the WHO essential diagnostics list. Right. And then from that same study, two important other findings as well. So only 23% indicated in achieving formal ISO accreditation, and only 15% reported using an electronic system, which is really important if you want to be able to export and transfer that data and use it for downstream analysis. So what are some of the, clearly resources and funding are major problems, but there are also other challenges that make this that cause this limited availability of testing. And notable ones are that temperature, humidity, dust, sunlight are not necessarily going to be the same as the conditions that Biomarot in France is or BD elsewhere in the US, or Copan in the US, those temperatures and conditions are not going to be the same as where these tests potentially are going to be implemented, because the resources and the funding available at those sites is not the same as implementing a Copan assay or placing a Vitek instrument in Europe. They may not be designed to endure those conditions. So, for example, on the right, you can see an example of development of condensation within glass slides because of the high relative humidity in Democratic Republic of Congo, right? And then it's also important to note that instruments may not necessarily be designed to account for intermittent or intermittently available electricity, where a instrument that consumes little energy might be optimal for a low resource setting. And maintenance is a major issue. So service engineers may just not be available in rural areas. And if an instrument breaks, the only way to repair it is to buy a new one or ship it to Europe or the US. And we've specifically encountered that issue within our Department of Chemistry, who have a specific mass spectrometer that has a flaw that could not be repaired via Zoom, and the only way to have it repaired would be to ship it to the supplier, because they don't have a service engineer that can come out and repair it. Then importantly, the appropriateness of local epidemiology. So best example I would say would be the antigen testing for Legionella pneumophila, where serotype 1 is predominant in the US and Europe, but not necessarily in South Africa. So while we may be sending out negative results for Legionella pneumophila, that may not be accurate. And we did find some cases when we evaluated a molecular assay. So those are specific challenges, but there are also measures that we can take to mitigate or try and reduce the relative or try and reduce the burden or improve the service of clinical microbiology in low resource settings. So, for example, things like rationalized bacterial identification and antimicrobial susceptibility testing. Not every organism we need to identify to species level. In some cases, knowing the genus is adequate and will not change management if further identification is done. And then importantly, also selecting susceptibility testing approaches relevant to available drugs. So you don't necessarily need a Vitek 255 card, which has a massive panel of antimicrobials, when there's only a few, when only a set number of drugs are actually available at the site. In those cases, it may be preferable to use a disk-based susceptibility testing rather than an automated method, for example. Improving communication between laboratory and clinicians, that's extremely important. Because especially if a laboratory is a low resource setting and may have frequent stockouts of specific supplies, or if there are going to be delays, but due to instrument failure with an engineer needing to come from needing to fly in from another country to repair an instrument, that can have significant impacts on clinical care if there's not constant communication between the laboratory and the clinicians. And that may, in fact, if that's lacking, then you may have clinicians moving away from doing tests, doing fewer tests, less data becoming available, less resources being allocated to improve infectious diseases care. Then prioritization is important. So it may be tempting to implement a susceptibility testing or identification for every possible pathogen, but in some cases you may only want to, for example, do the high priority tests like blood cultures, where, for example, even CSF, because of a relatively small set of common pathogens with very predictable susceptibility profiles, microscopy, gram stains, and cell counts may not add that much benefit over. Adding culture to that may not add much benefit. Certainly, adding molecular testing to it would. But if the decision is just, do we add culture or do we add blood culture? How do we prioritize it if we only have the capacity to offer a set number of cultures each day? And then generating training materials dedicated to the setting is really important. Making sure it's available in the local language, is open access, and is updated to reflect current practice or current best evidence is important. To do that, there are various tools that are available. So the WHO has released, for example, the SLIPTA checklist, so a stepwise laboratory quality improvement process towards accreditation, which is designed to be a biannual site audit of laboratory procedures such that a laboratory can work towards becoming accredited. And you can see here below as an example of, for example, elements that would be in the checklist that would be completed each year and be used to guide improvement. And then companies have also released things like the mini lab that the MSF uses, which is essentially a fully functional microbiology lab in 3 crates, which includes a laboratory information system, documentation, reagents that have a long shelf life, microscopes, incubators, autoclaves for waste management, and laboratory benches to protect staff. And that can be set up in 2 days with a month of training required, followed by essentially mentoring activities. And this can be deployed very rapidly to a site that requires a medical microbiology service. The notable issue, though, is that it has limited capacity. This is not a long-term permanent solution. You can do maybe 10 blood samples per day, possibly expand to around 20, but it is certainly not a solution that you can implement in a lab and that make this your permanent lab. So it's really a system that can be used to drive further access, that can be used as a basis to expand the service in a more sustainable way. Right. And then really there's the leapfrog solutions, if you will, where diagnostic technologies where you can bypass the infrastructure requirements of systems that are implemented in high income countries. So that would be like point of care, affordable diagnostics, especially ones potentially that could distinguish between viral and bacterial infections, or identify pathogens and provide antibiotic susceptibility testing. So the best example of that would be, for example, the Cepheid cartridges for MTB, which can provide both identification and susceptibility testing, and essentially have temperature stable cartridges, as well as the entire system being contained in a cartridge, which can then easily be disposed of as well. Notably, these tools need to be adapted to low middle income country constraints. They would need low maintenance needs and be able to handle varying heat and humidity levels. It's certainly not useful to have a very impressive molecular assay, but you can use it twice and then it breaks and the engineer needs to take 2 weeks to come out to the site to repair it. And yeah, that's my talk. I hope I'm within time. So there's still time for questions.
Thank you very much, Ruan. Yes, definitely some time for some questions. So I would open the floor and everyone can put their hand up if they have a question, or just pop it into the chat box and we can read it out loud. Graham, your hand is up.
Yeah, thanks so much, Ruan. Really great overview, and really, for me, illustrated the complexity and the multifaceted nature of addressing this problem. And clearly, to scale up microbiology services across Africa at each hospital is a huge long-term effort and with health system strengthening. But I'm just wondering, as a first step towards better surveillance for AMR rather than necessarily being able to provide it in every facility or for every facility, what initiatives are there underway to kind of get a better handle on AMR within countries at sentinel labs, particularly in low-income countries?
So the WHO has produced, for example, standards like I think it's called GLASS, which provides, for example, standard like data points that need to be collected so that the data is comparable. Then there's plenty of like NGO work that focuses on specific pathogens. So, for example, I work with the Global Alliance for Preventing Pandemics, where their focus is obviously with my interest being sequencing. They implement increase access to kind of sentinel sequencing services where you can basically produce primarily virological like whole genomes for epidemiology. But yeah, I mean, one of the main aspects, like as you mentioned, is and I think that I put it on a slide, but didn't necessarily mention it, is basically, oh, no, not there, but basically effectively not just rationalizing your identification and susceptibility testing at site, but also rationalizing what testing is sent away and what testing is kept local. So do you need potentially the susceptibility testing for the organism, but you don't necessarily need the typing, though you do need the typing to monitor what vaccine strains to use for your drug, for your vaccine. So that kind of like national reference laboratory outlook of pairing with this regional lab so that things are not duplicated, I think would definitely be the primary way to go. But other than that, essentially utilizing the tools that are available, like SLIPTA, like the GLASS framework, to kind of guide which are the parameters that are going to be useful for integration of LMIC data into these larger studies that ultimately drive policy.
And is there large-scale funding being provided for that kind of surveillance work across low-income countries?
So, I mean, my impression, and I mean, I'm certainly not maybe not the best person to ask here, but I mean, Wellcome does provide a lot of funding focused on surveillance. I mean, there are certainly grant calls from EDCDP, especially that I have looked at or considered applying, that are really often focused on then a specific clinical syndrome. So I think last year's call was they had a focus on gastrointestinal pathogens. So there certainly is funding available for that type of work, but it is often really for a specific disease syndrome or specific pathogen. And yeah, I mean, I don't think I'm necessarily on the right national boards to be aware of the larger scale funding.
Yeah. I don't see that there's any other hands up yet, but maybe, Ruan, I can follow up with a different question, which is right at the beginning, you showed the mortality estimates for AMR across different continents. And it seemed that Africa was the estimates were highest for mortality related to AMR. And I find that quite surprising because, in high income settings, there's obviously lots more hospitalization, use of far wider spectrum of antibiotics. And, people are, often in hospital, in ICU, far more ICU provision, people on immunosuppressants, that sort of thing. So what do you think accounts for the very high rates of mortality due to AMR in Africa? Because it clearly isn't hospital-acquired infections like in North America and Europe, for instance.
So, I mean, I think it probably will be granular in terms of specific factors of specific countries, but potentially, like the issue may well be that. When looking through the supplementary data for this paper, I mean, the thing that mostly stands out is that most of the data from Africa is from like clinical trial sites and from academic centers rather than community data. So it may well be that the cases that you are capturing there are that kind of subset who have access to drugs that might be available in higher income countries, but don't have necessarily the ICU infrastructure, and the other support infrastructure necessary to deal with severe infections. And potentially, when those individuals reach care, you might have delays in starting appropriate therapy because of delays in antimicrobial susceptibility testing. So you might, for example, start with because of, you might have, let's say, lack of surveillance data, so your empiric regimen might not be appropriate to your current ESBL rate. And you might only find out, from the laboratory that you have an ESBL when the patient is far less well. So I think it's a combination of those things. And I think probably the data would be different if you also had community data from Africa. But I mean, I don't know, especially particularly because that just isn't available. But yeah, I think the main things would be, this is a biased data set, in a setting with potentially access to testing via it being clinical trial sites, but potentially not access to the same level of supportive care that you might have in high income countries.
Yeah. Thanks very much, Ruan.
I still don't see any hands, and there are no questions in the chat as yet. But please feel free to email us afterwards if you have any, and we can always share it with Dr. Marais to answer. His talk will also be available on the Revive website with his permission, and his slides will be added there. So if there's nothing more from the floor, I would just say thank you very much to everyone for joining, and I wish you a very good week further. Thank you. Thanks, Ruan.
Thank you. Bye-bye.